During this session, I'll walk you through how to access the tool from rcsb.org, the types of alignments it supports, and how to interpret the results and visualizations. We'll also have time for your questions at the end, so feel free to use the Q&A feature in Zoom to ask them as we go. Before we jump into the tool interface, I want to briefly introduce what pairwise structure alignment is and why you want to use this tool. Pairwise structure alignment is a fundamental technique in structural biology for comparing the three-dimensional shapes of two biological macromolecular structures. And unlike sequence alignment, structure alignment focuses entirely on the 3D arrangement of atoms. It works independently of sequence identity and ignores the specific types of amino acids when deciding which parts of the structures correspond to each other. And on RCSB.org, the pairwise alignment tool makes this process very straightforward. The tool has a simple interface. It allows you to align one or more protein structures onto a reference structure, as well as visually and quantitatively compare them side by side. Let's talk about why you might want to use it. So researchers often turn to pairwise structure alignment when they want to identify structural similarities between proteins they don't share much sequence identity. Structure can be conserved even when the sequence has changed dramatically. It's also useful for detecting conformational changes between different functional sites of the same protein, exploring evolutionary relationships, or comparing experimental structures to predicted models. And finally, it is a valuable way to validate your own modeling or refinement work, making sure that the structure you build matches what you expect. Let's take a look at where you can access the tool. The easiest way to start is from the top navigation menu of the rcsb.org website. You locate the Analyze panel and select Pairwise Structure Alignment. And this will open the tool interface where you can select what structure you want to compare. Let's take a look at the website live. So this tool has several options to specify which structures you want to align. By default, you can enter either PDB ID or computer structure model ID if you know them. This allows to look up existing structures on the PDB archive and more than 1 million predicted structures from AlphaFoldDB and Model Archive that we incorporated into our website. You can also use a UniProt accession to fetch structures associated with a particular protein sequence. So this drop-down allows you to inspect what known protein chains have 3D structure and are deposited either in PDB archive or available amongst a million computer structure model that we incorporate. So for predicted models, the tool lets you to pull directly from AlphaFoldDB or ESM Metagenomic Atlas using their identifiers. And you can also have an option to upload your own coordinates, either by uploading a file in one of the accepted formats or by referencing the file hosted elsewhere by pointing to a URL where this file is located. So this makes the tool powerful for comparing experimental data, predicted models, or your own structures all in one place. There is a number of algorithms that we provide to perform structure comparison, and I'll cover them in detail in the next slides. Now let's take a look what you'll see once the alignment is performed. The tool provides both visual and quantitative results to help you interpret the comparison. So first you will see the key metrics in this top panel that quantify the alignment. For example, you will see an RMSD or root mean square deviation. It measures the average distance between aligned atoms and tell you how closely the structures match overall. Lower values indicate better alignment. TM score is a normalized score that takes into account protein size, and it ranges between 0 and 1, where 1 indicates a perfect match and 0 is no match between two structures. Typically, scores below 0.2 indicate that the proteins are unrelated, and the scores that are greater than 0.5 generally have the same protein fold. You also see the sequence identity that shows the percentage of residues that are identical in the aligned structures, and the alignment length, which measures how many residues were matched between the two structures. And you can compare it with the full sequence, deposited sequence length, and the number of residues in the structures that do have corresponding coordinates. Below that, on the left side, the tool provides detailed residue level mapping, so you can see exactly which part of the structures correspond. And on the right, you'll see a 3D superimposition of the aligned structures in the interactive viewer, letting you visually assess how well they overlap. Those two panels are connected in a bidirectional way. So when you highlight region in one panel, the corresponding region in the other panel will be highlighted as well. You can also download the alignment report and the files with coordinates that are transformed to superimpose structures onto each other. If you want to download those files and use them in other software for further analysis, you can do that with those expert options.
Hello. Hello.
If you could please mute yourself, questions can go into the Q&A button. Thank you.
Let's take a closer look at the alignment algorithms supported by the tool. So first, we have several rigid body alignment methods that treat the structures as fixed shapes during comparison. TM align is the default method used by the tool, is a fast method. It uses TM score metric and best suited for proteins with similar overall fold. Fat cat in rigid mode is another option. It identifies the largest conserved structural core by superimposing structures without allowing for flexibility. CE, or combinatorial extension, searches for the best matching substructures by building alignment from small aligned fragments. And Swiss Waterman in 3D, it's a sequence-dependent method. It uses sequence alignment to inform structure superimposition, and it works best for comparing closely related proteins with very high sequence similarity. On the other hand, we have flexible alignment method that can account for domain movements, structural rearrangements. For example, fat cat in flexible mode introduces twists between different parts of the protein so that it can be aligned independently. It allows detection of internally rigid domains even when large conformational changes occur between structures. CECP stands for combinatorial extension with circular permutations. So it's designed to compare proteins with similar overall shapes, but different connectivity due to circular permutations. For example, when an N-terminal part of one protein is related to the C-terminal part of the other protein. Understanding which algorithm to use depends on your specific needs. For example, if you expect rigid or flexible differences between your two proteins. To illustrate how those alignment methods work in practice, let's take a look at those examples. So first, we'll look at rigid alignment between mammalian tubulin and bacterial FTSZ protein. So these proteins are homologous and they play critical roles in cell division. And despite their very low sequence identity, you can see that the structure similarity of these proteins is pretty high. Next, we'll take a look at flexible alignment example. We will compare two Calmodulin structures, calcium-free and calcium-loaded. So Calmodulin has two lobes, each with EF hand calcium binding motifs. In absence of calcium, these EF hands adopt a closed transformation, which opens up upon calcium binding. You can see well the differences in this conformation if you apply a rigid body method that does not allow aligning different parts of the structures independently. So the calcium free structure that adopts a closed conformation looks like this. And here is the calcium loaded structure in the open conformation. So with the flexible method, you can capture the alignment of those conformational changes more effectively. And finally, a circular permutation alignment between concanavaline A and peanut lectin. Both are plant lectins with similar tertiary structures, but different linear sequences. Their N and C termini occur at different positions in the structure. You can explore that in the sequence viewer, for example. Here you see the sequence numbering that shows how an N-terminal of one structure corresponds to a C-terminal of another structure. Again, if you choose to apply a rigid body method, you can see that only half of the residues can be aligned compared to a method that allows for flexibility and circular permutation detection. The next example showcases one of the more advanced uses for the pairwise alignment tool, aligning multiple experimentally determined domain structures to a full-length computer structure model. Let's take a look at this example. So often full-length experimental structures are not available, especially for large multi-domain proteins. However, individual domain structures determined experimentally can be aligned to full-length predicted model. In this example, we're using an AlphaFold model and two available PDB structures. So this approach allows researchers to build a composite view of understanding of the entire protein's architecture by combining the high confidence domain data with computational predictions. You can also see that the partners in the complex, if they're available with experimental data, can be placed alongside the alignment of the protein chains as well. An important point to highlight is that the pairwise alignment tool you use through the rcsb.org website is backed by a publicly available API. This means you can programmatically access the same alignment computation that are used in the UI, but programmatically incorporate them into your own workflows. Before we wrap up the presentation, I want to point you to several resources that can help you get the most out of the pairwise alignment tool. So you can access the tool directly at this URL. There are detailed instructions and examples in the user guide that is available as a link through the tool interface. If you use your tool in your work, please cite the published paper linked here with this reference. You can also find this reference on the interface page of the tool. We have tutorials and references for API with detailed explanation. And finally, I want to mention that the UI uses open source components, which include the sequence annotations tool that is used by the alignment interface to display sequence alignment. Those are public GitHub project, and you can find those projects on the GitHub. And finally, the full pairwise alignment UI implementation is also publicly available on the GitHub as well. Thank you so much for your time and participation. If you have any questions or feedback after the session, please don't hesitate to reach out to us at info@rcsb.org. You can also use our website for more information, tools, resources, and contact us via this Contact Us button. Before we go to a Q&A session, I want to highlight the upcoming training opportunities. The webinar recordings from previous sessions are posted at PDB 101 at rcsb.org, which you can watch anytime. If you're interested in the future training events, I encourage you to sign up for our mailing list to receive email notifications about upcoming webinars and workshops. And looking ahead, we have an upcoming webinar titled Searching the PDB for High Quality Ligand Bound Structures, scheduled for Monday, September 8. We hope you can join us for this informative session. Also upcoming is the virtual office hour focused on PDB policies for deposition and biocuration on Wednesday, September 25. This session is a great opportunity to engage with the expert and ask any questions you have regarding deposition and biocuration process. Also, as a reminder to the community, the traditional 4-character PDB IDs will become fully allocated by 2028 as the database grow. To address this, PDB is transitioning to extended PDB identifiers, which provide a larger namespace to uniquely identify new structures. And going forward, when we switch to those new identifiers, model files will be only provided in MMC format, not in the legacy PDB format. So we encourage users to become familiar with this change and the MMC format as soon as possible, because it will become the standard. Thanks again. Now, if you have any questions, please use the Zoom Q&A feature to submit them. And I'll do my best to answer as many as the time allows.
Thank you so much, Jana. And Shamar, you can stop recording. Juwan?